Hello friends, today we are going to study about protein function in which complementary interactions between proteins and ligands. In the last few videos, we were studying about the function of the protein involving a reversible interaction with their ligands. In this lecture, we'll study about complementary interaction of proteins with ligands. Now, the immune system of our body, or we can say the proteins of immune system of our body function by complementary interaction. Now, what is immune system? Immune system means it is the system that provides protection. Protection from germs or any foreign particles that can harm our body. So, it is very important for the immune system first to distinguish between self and non-self. That means which is the molecules that are of our body and the molecules that entered from external environment. So this discrimination is very essential for the immune system. After discriminating the non-self parts, it should kill that non-self part to protect our self part. Now, for this killing, there occur a coordinated set of interactions at physiological level. So, the immune system is the system that protects ourselves. For the protection, it needs to distinguish between self and non-self. And after recognizing the non-self, it should kill the non-self. And to kill the non-self, it is a set of coordinated interactions at the physiological level. Now, what are the cells and the proteins involved in the immune system? Now, because we'll study immunology in depth in the immunology subject, in this lecture, we are just getting the overview of the immune system. So, there are multiple cells and proteins involved in the immune system, but we can divide the immune system in two sections, that is humoral and cellular. Humoral consists of mainly antibodies, and these antibodies are produced by B cells. Coming to cellular, it mainly consists of T cells. I'm using the word mainly in both the cases because there are much more cells and proteins involved in the immune system, which we will study in the immunological subject. Now, T cells are of two types, TC and TH. TC means cytotoxic. That means it will kill the foreign substance. TH means helper. It will not kill directly, it will help. Whom it will help? It will help TC cell, it will help B cell to produce antibody. It can even interact with macrophages. What are macrophages? These are also the cells of the immune system that phagocyte the foreign particle. They kill by phagocytosis. That means they eat and then digest. Now, how these T cell will recognize the non-self part? Through a receptor on their surface, which is known as T cell receptor. So T cell receptors, these are cell surface protein which will bind with the ligand, and then a signal transduction process will start for the action. So immune system have two sets, humoral and cellular. In humoral, the main is the antibody produced by B cell. In cellular, the main is the T cell, having main two varieties, TC and TH, where TC means cytotoxic, they'll kill the foreign substance, and TH means helper. So they will help TC cell to kill, they will help B cell to produce antibody, they will help macrophage cell to phagocyte. Now, how T cells will recognize the foreign substances? Through T cell receptors. Now, what are T cell receptors? These are the receptors that are on the cell surface. Here the ligand will bind, and that's how a signal transduction process will start and the action will be taken. Now, self and non-self discrimination. So the self and non-self discrimination is done by MHC, that is major histocompatibility complex. Now what they do, they will digest and present the peptide on the cell. So suppose this is a MHC. So what they will do, if any foreign substance will enter, they will break this substance into smaller peptides, and then maybe a section of this peptide will be displayed on its surface. So as soon as any peptide being displayed on MHC molecule, it will be treated as non-self. The body or the T cell specifically will consider these peptides as a non-self or these proteins as a non-self which is displayed by MHC molecule. So this non-self particle or the peptide will then recognized by the T cell receptor present on the surface of T cell. So that's how any foreign particles or any self particles that need to be damaged is presented by MHC molecule and get recognized by T cells. Now, MHC molecule come under two classes, class 1 and class 2. Class 1 MHC molecule is present in almost all cells and they display peptides that are degraded within the cells. This will, after presentation, will get recognized by TC cell, which will then destroy this cell which has got infected by parasite or any virus. Class 2 molecule, these MHC molecules are displayed on the surface of macrophages and B cells. So basically, macrophages and B cells are the cells that take up external particles. They will then display these peptides and these will then get recognized by TH cell. So MHC class 1 will work to recognize the cells of our body which get infected due to parasite and virus. It will recognize that cell and help in killing that cell with the help of TC cell. What will class 2 MHC will do? If any foreign particles get entered in our body from outside, it will take up that peptide and will present to TH cell. Now, next function is TH cell. It is a helper cell. So it will then proliferate or it will then help TC cell or B cell or macrophages for the killing of these foreign particles. Now, in our blood, T cell will patrol. It is a patrolling team who will just patrol in the bloodstream and the tissues in search of foreign particles. As soon as they will encounter any foreign particle, they will launch subsequent steps for immune response. Now, antibodies. What are antibodies? These are anti to the bodies. And bodies means not our body, it is antigen. So whatever anti to the antigen is antibody. So antibody will bind with the antigen and mediate its killing. Now how the antibody recognize antigen? Because all the antigen will have some antigenic determinants. That means the mark that helps the antibody to recognize the antigen. So all the antigens will have some determinants which are also known as epitopes. So antibody will bind with these epitopes and this binding will only work for creating memories. So in the next time when these antigens will enter from natural environment, these antibodies will recognize it and will mediate its killing. And this interaction is also responsible for immediate killing as well. So this interaction is very important because with this interaction only the antigen will get recognized by antibody for present as well as for future encounters. Now antigen is also protein, antibody is also protein. That means this is a protein-protein interaction. And when this is a protein-protein interaction, then how these two parts is interacting? So there should be some complementarity in shape. Then the interactions will play an important part because these are amino acids. Protein means these are made up of amino acids. So depending upon the character of amino acid, is it a polar amino acid, non-polar amino acid, charged amino acid, the various interactions will come into play for binding of antigen and antibody. Now, how the structure of antibody looks like, if you can see in the structure, V stands for variable, H stands for heavy chain. If you see, this chain is very long, having VH, then CH1, then CH2, then CH3. It is a long chain, hence it is heavy. Beside that, we have VL. V stands for variable, L means light because it has only two segments, VL and CL. C stand for constant. So these variable regions are only responsible for binding with varieties of protein. Now, this heavy chain and light chain, you can see these are attached with disulfide bonds. And this region, the center one, it is known as hinge region, from where the antibody is made flexible because they should have complementarity with shape. So that means if the antigen shape, for example, is like this. So the antibody should be like this. Now, if the shape and size of the antigen is like this, the antibody should open its arm more. So for this flexibility, it needs a hinge region. So this center part made this circle green center part, this is known as hinge region, which provides flexibility to the arm of the antibody. And this is also stabilized by or connected by disulfide bond. Now, what else we can see in the structure? This antibody is having one amino terminal. You can see the arrow. This is amino terminal, and it also has one carboxyl terminal below, COO minus. So towards the amino terminal, it binds with the antigen, and with the carboxyl terminal towards this terminal, it remains attached with the cell, that is B cell. So antibodies, they will bind with the antigen through their variable region. These are variable because so that they can bind with varieties of antigen. So these antibodies are very specific for their antigen. So the specificity has been maintained due to this variable region because all the antibodies will have different variable regions. So that's how an antibody will recognize the antigen specifically and it will bind. Now there are various classes of antibodies: IgG, IgM, IgA, IgE, and IgD. IgM, it is the first antibody that is generated in response to any antigen, and it is found in bloodstream. IgG, it is the most abundant antibody of the bloodstream, and IgG is the major antibody of secondary immune response. Secondary immune response means whenever any antigen will enter, the body will release IgM. But when the same antigen will enter second time, it will release IgG. So it is the major antibody of secondary immune response. IgA, it is found in secretory fluids like saliva, tears, etc. IgE, it is used in allergic responses or it creates allergic responses. And IgD, it is used as a marker. So these in short about immune system where protein function through complementary interaction with its ligand. This is just a very brief overview of immune system. We'll study in depth about complete immunology as a separate subject. So now one question from my end for you all. You have to tell me which amino acids are found in the hinge region of an antibody. So what I said, that disulfide bond is present. Hinge region is also responsible for providing flexibility. So keeping these properties in mind, I guess now you can easily tell that what kind of amino acids can be found in the hinge region. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe to the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.